everyone! Welcome to our next virtual lesson, Coral Restoration, Growing Coral on Land. There are no materials required for this lesson. However, you may want a pen and pencil, a notebook, or maybe it will just inspire you to grab your mask and fins and go for a swim after this. For our learning objectives today, we hope that you discover, understand, and identify. Discover what an ex situ coral nursery is, understand the advantages of land-based coral nurseries, and identify some techniques scientists use to help coral grow faster. Let's dive in. Coral restoration began in the water with in situ nurseries. Corals are grown on coral trees like the ones you see in this picture. These trees are suspended in the water, which gives the corals lots of light, nutrients, and water flow. Corals love to grow in these nurseries, and staghorn coral, which is what we're seeing in the picture here, grow fairly quickly, for corals that is. The only real disadvantage to growing corals in an in situ nursery is that getting to the nursery can be weather dependent. We're taking boats out, we're going scuba diving, so we have to make sure that the conditions are safe for our divers to go out. If you want to learn more about in situ nurseries, we already have a lesson about it and a behind the scenes video on our Google Classroom. You can find the information about how to join the Google Classroom and the code at the end of this video. So in addition to our in situ nurseries, Mote has an ex situ or land-based nursery. This is a picture of our facility on Summerland Key in the Florida Keys. You can see several blue tanks with black lids on top next to the building. So these are actually a part of our ex situ or land-based nursery. We have additional tanks not pictured here, as well as indoors in our wet labs. So if you're getting confused about in situ or ex situ, to help you remember, think in situ, in the water, ex situ, ex it the water. So here in this picture, we're looking inside of a raceway. Raceways are the tanks used to house and grow corals for research and outplanting. We grow two types of corals in our land-based nursery, branching corals and boulder corals. This branching coral you see here is an elkhorn coral, and we do a lot of our land-based restoration working with this type of coral. It's characterized by frond-like or flattened branches, and these corals used to be a dominant coral on the Florida Reef Tract, but they are now listed as threatened on the Endangered Species Act. Boulder corals are much slower growing and create large mounds on the reef. And both of these types of corals are important for building the 3D structure of the reef. What you are seeing here is a raceway with racks of individual plugs holding corals. We can take a parent coral, about the size of a dinner plate, and create hundreds of little individual fragments consisting of individual polyps and allowing them to grow in our land-based nursery. Individual corals are attached to hard ceramic plugs and placed on racks like this. And we can have thousands of these, up to 40,000 at a time, in our land-based nursery. We use special techniques called microfragmenting and reskinning as a way to restore the reef. Scientists use diamond-bladed saws and take half-dollar-sized corals called medallions and cut them into 1 square centimeter pieces. You can see an example of this in the top right-hand corner. This technique is called microfragmentation. But if we're trying to get corals to grow bigger and faster, why would we cut them smaller? Cutting the corals into small pieces creates a growing phenomenon. For example, if you have a coral, let's say the size of a softball, it can grow about 0.1 centimeters, or think of the size of a pinhead over a few months. But when you microfragment them, you can actually accelerate the growth rate, which can be up to 40 times faster on average for the species we've worked with so far. Taking these corals and cutting them triggers this growth response similar to our skin. Our skin cells don't grow very quickly normally, but when we are cut, it triggers the cells to grow much faster in order to heal. And that's exactly what we're seeing here with microfragmentation. And corals cut from the same medallion can actually fuse together. This means if I put two corals next to each other, when they grow out and touch, they can connect and then continue to grow as one coral. And that's because corals cut from the same medallion have the exact same DNA or genotype. And this video shows corals fusing together really sped up. In our land-based nursery currently, we have about 27,000 fragments, 13 different species of corals, and 813 genotypes. And it doesn't stop there. Once our corals have grown out to the medallion size, and before we put them back onto the reef, we are testing different genotypes for resilience. We want to make sure that we're giving our corals a fighting chance. Moat scientists work with different genotypes from each species and expose them to the 3 most important threats identified for corals in the Florida Keys. Here we can conduct more informed restoration activities and strategically place these individuals within reefs where they are most likely able to survive in the future. The 3 most important threats are disease, high water temperatures, and ocean acidification. If you're interested in learning more about these things, we will be having additional lessons coming out very soon. After corals are grown in the nursery, tested for resilience, now it's time to get them back out on the reef. 90% of our land-based corals are transported underwater for outplanting. We keep about 10% of these corals to continue microfragmenting and propagating our land-based nursery. If you remember what we talked about in the video where the corals were fusing, it's important to plant corals with the same DNA close together. Otherwise, they'll compete and fight and they will not fuse together. You can see here that we have a dead coral head, and you can see different arrays of corals that we've planted, basically in kind of a circle on a dead coral head. And now over the next few months, they will continue to grow outward, touch, and fuse together. This technique is called reskinning. In 2018, moat scientists outplanted 4,880 outplants from our land-based nursery. And in 2019, they outplanted 5,083 outplants from our land-based nursery. After outplanting, we monitor our corals to find out how they're surviving. And we measure survival by attachment success, individual coral survival, and the array survival. In this first graph, we're seeing data from our elkhorn coral survival and success. Attachment success ranges from 97% at 1 month and 92% at 1 year. Individual coral survival ranges from 91% at 1 month and 98% at 1 year. And array survival ranges from 98 to 100%. Mortality is usually driven by predation, fish and snails. But overall, this is a huge success for our outplanted corals. This next graph we're looking at is our boulder corals, and it's a little bit more complicated. So we can see that attachment success ranges from 90 to 99%, but the individual coral survival ranges from 1 month, 6 months, and 12 months at each different location. Offshore, mid-channel, and near shore don't look to be so great. But this is actually because some of these corals are used for research and not just restoration. And a lot of these corals were a part of a research project that was looking at disease susceptibility in boulder corals related to a newer disease called stony coral tissue loss disease that's been present in the Florida Keys since 2014. Overall, we can see that our nearshore corals, though, have a lot of success from 100% at 1 month, 93% at 6 months, and 90.4% at 12 months. In conclusion, land-based nurseries allow moat scientists to use cutting-edge technologies to grow and to study corals. Using microfragmentation, scientists can grow corals up to 40 times faster than they can grow on their own. And outplanted corals used for restoration have a high chance of survival. If you want to learn more about microfragmentation, come behind the scenes with us tomorrow. Moat scientists will be demonstrating microfragmentation in our next behind the scenes video. You can find all of this content on our Google Classroom. Information about how to join will be posted at the end of this video. We want to thank the NOAA Restoration Center Award for funding these education opportunities as well as our restoration work here in the Florida Keys. Thank you guys so much for joining us today. We hope you enjoyed it, and go ahead and leave us some feedback. Let us know what you think, and we hope to see you tomorrow.